Urban ecology of orb-weaving spiders, the effect of noise pollution on the foraging behaviour of Zygiella x-notata. Rapid urbanisation is one of the effects of population growth and development. It's one of the drivers of pollution, and for this poster, I'll be focusing on noise pollution. Noise pollution is caused by things like foot traffic from people, construction noises, as well as motor traffic. Increasing background noise levels in cities is something that many taxa have had to adapt to, and I am particularly interested in seeing how spiders do so, given that so much of their lives are built around vibrations. Their webs are an extended phenotype used to interface with the outer world, not only to encounter mates and trap prey, but it has also essentially evolved to act as an antenna. So given that spiders have evolved to interface with their surrounding soundscape, how does the change in atmospheric noise affect their ability to build webs or forage? I focus on the orb-weaving spider Zygiella x-notata, chosen for its abundance in Oxford, especially in noisy roadside locations. My experiments took place in two parts. The first was to measure different parts of the web and compare those across quieter green park sites and noisier urban sites. So I measured things like the number of anchor threads, radial and spiral threads, and as well as measuring the length of things like signal threads and different size metrics using digital calipers. These measurements were also used to calculate parameters like capture spiral area, mesh height, and asymmetry. In the end, I recorded 98 different webs across both sites, 46 of those being in parks and 52 from urban sites. The second part of my experiment involved recording spider reactions and responses to simulated prey frequencies. I recorded videos of me holding tuning forks at 34 different webs across both sites, half of those being from park spiders and the other half from urban. I used video processing software to find the reaction times and classify different reaction response types. Tuning forks have been used for years in lab settings, but recently their proficiency for field experiments was shown by Davies and Hesselberg. I used 4 different frequencies, with the highest, 440, being used to simulate small wingbeat frequency from common spider prey, and the lowest, 32 Hz, and it being an experimental frequency used to simulate ground level vibrations that come from traffic. The other frequencies, 256 and 128, were used to simulate larger prey or predator species. My results found 3 different findings. The first was that there was no difference in web construction depending on the amount of noise in the environment. So here's a graph showing the different parameters of the web, and that there's no significant difference between those found in park areas, shown in blue, and in urban areas, shown in orange. Secondly, I found that there's no difference in reaction times or response types to tuning fork stimuli between sites. Again, the park sites are seen in blue and the urban in orange. And lastly, there was no difference in the proportion of responses from different tuning forks between the sites. In this graph, you can see the park sites in the top row and urban in the bottom. The different colours represent different types of responses, and there was no significant difference found between different forks or between different sites. These results were rather unexpected considering previous findings for tuning fork experiments, for example those done by Davies and Hesselberg. They found in their paper that there was significantly more attack responses for 440 Hz compared to 256. This difference in results could be for a variety of reasons. It could be that other factors had more influence on web construction, such as prey size and availability, as well as space available to build a web. It could also be because the 2022 paper did not use Zygiella x-notata, so further work could be done to find out if tuning fork experiments work equally on all species. Though there were no significant results here, there were some trends observed, such as in the pie chart, there are a slightly higher proportion of no reaction and non-attack responses in park sites compared to urban sites. This could be because urban green spaces harbour more insect and bird life. The findings in this paper could make sense given the ecology of this species, which is known to thrive in urban environments, which is quite remarkable given that the selection pressure of noise pollution could not have existed for more than 200 years, and it could be interesting in further experiments to find out whether this adaptation is genetic or plastic. It would be interesting to explore further how humans can have such an impact on the evolution of species that coexist with us.